Great. So just to maybe give a little bit of an overview of things here. So this course explores the basics of biological microscopy analysis, and we will start by looking at, first of all, what is an image, what is a bioimage, and then just the concepts around that, like just what you need to know about where image comes from, what is actually stored there, how do we actually work with these intensity values, etc. And then from that we will then say, cool, now that we understand how these intensity values are put together, how can we visualize this in a way that is helpful for our analysis? And then the final step is then to actually analyze it, to extract some meaningful biological parameters from the images. And then just some things to consider, like for example, if you change the intensities of your image, then the intensities are no longer directly meaningful for your analysis, so just be cautious of those types of things, like what you do in the pre-processing affects your analysis. So some of those nuances we'll just look at as well. Maybe just the software, you would have all known this by now, but we will be using Fiji as a base for this, but there are many alternative pieces of software that can do almost the same things. Fiji is obviously a free, great open source, massive community tool, and that's why we're using that. So I'll be coming back to this picture a few times, but the rough flow or pipeline for any analysis is you start with your biological problem. Now, you've mentioned the problems that you're working with, but this is a very important first step, is to actually define what is the thing that I'm trying to see, what is the thing I'm trying to get out. I've got certain hypotheses. Can I just look at an image initially, and then can I kind of see that in the image I get that information? So it starts with what is the problem, what is the actual thing I want to quantify or analyze or visualize or whatever, and then you just first do a gut feel, you get you acquire some images with a microscope, and then you say, okay, well, can I with my eyes hint at what I'm trying to get at? Is the information actually there from the raw pixels? And then it's like, okay, it seems like this is reasonable, a microscope has got enough resolving power, all of these things that we require, and then now we start with the actual analysis and visualization. So in the visualization section, there's some pre-processing and enhancement that you can do. Sometimes the intensities are quite low, you want to boost them, etc. And then you want to often isolate the different objects that you're trying to analyze. So for example, in mitochondria, you might want to get the different mitochondrial structures that are in your cell. I don't know, let's just say another one. The nucleus, you maybe want to count nuclei in your population or something like that. So you first need to segment or separate out, this is the structure that I'm wanting to investigate. And then from that, you can then extract the data. So either sometimes the morphology itself is interesting, so there you just use the segmented image. Other times you actually need to look at the intensities and the variations in that, so then you take your segmentation, align it with your intensity, and then measure some parameters there. And that's the kind of last step where you do features and parameter extraction to then ultimately, I suppose there's a last step, which is then the statistical analysis on those extracted parameters. Okay, so from this biological framework, let's just use an example. This is not my example. This is, I think, my colleague Emmanuel that presented the course in Uganda's example. But let's take this flatworm, the planaria, I believe it's called. It regenerates itself if you cut it in around 1 week. And the question is, are there some differences in terms of the contraction frequency depending on the size of the tail cut? So you've got your little worm there, you cut it somewhere, and then eventually it will be formed now 2 worms in this case. But you want to know in this, it's going to have this kind of contraction pulsating thing going on. And so if you image it, you see that it was pretty brief there, but you see that it stays a little bit stable and then it pulses. And you want to know, does this pulsating thing or this contraction frequency, is it dependent on the size? Can I actually now quantify, okay, if you were to cut it longer, is it going to pulse quicker or slower? And what's not written here is you always obviously want to know why do I want to say this? Like I've got now my problem here, but what is the point of getting this frequency? Is this frequency going to tell me something about the rate of growth? Is this going to tell me something about the, I don't know, some initial healing that needs to happen within this worm that's cut? So the big question of the why, what am I doing, why am I doing this, what is the reason why I'm wanting to get this? Because all of those reasons or questions will ultimately feed into how I'm going to do the analysis. Okay, so now we've got the raw pixels, then we need to start with some image processing. So we have our population of things that we want to analyze, and then we maybe identified 2 separate I don't know, parts of this worm that we want to know, analyze. And then we have, for example, a big one and a small one, and we can look with our eye and we can see, okay, well, we can see the frequency of pulsation that's going on here, but now we want to just model this statistically, that it's not just our eye, or we don't want to manually go and count through the frames, okay, how many frames did it take? We want to have an automated way so that we can do this on high throughput data settings, where we have lots of these things, and we just put it into some pipeline, we click run, and then it gives us all of these frequencies out so that we can do very robust stats on it. So okay, now we've enhanced it, we've made these selections around it, and let's say we look at the perimeter, so, or in this case the area, doesn't really matter, both of them would give you the same type of information, and then we can say, okay, well, over time, how does this pulsing happen? And then you can see, okay, there are these periodic pulses that happen, so then you just take, for example, from you can either do this for all of the peaks and then average it, for example, or say how many pulses happened within a certain time frame, and then you can get it from that. Or you can even do a Fourier transform and then turn this into a frequency domain representation, where you then say, well, where is the highest frequency? That is probably the frequency of, I can get the period from that of the contraction. And then you can plot it out for, you might want to separate your entire population into two separate things. There's a big one, there's a small one, and there's a lot of big ones, a lot of small ones, and then statistically speaking, what is the period variation between that? So for example, the big one seems to have a longer period of contractions, with the small one has a lower period of contractions. So yeah, this entire pipeline that I just went through is basically what you do in any problem. This is just a quick, dirty example to give you a feel for the type of things that we will be doing, and in the next day and a half or 2 days, we will go through various examples and look at each of these steps and try to give the tools as to how do you do this. Okay, so image processing. Maybe let's just define this first of all. So image processing is the process where you take digital images, push them through some algorithm, and then get some results, obviously, from this algorithm. Now, these digital images can have various sources. It can be from a camera, a normal photo camera. It can be from medical imaging devices such as an MRI or a CT scan. It could be from satellite imagery where you look over the Earth. It could be from your microscope. And all of them have something in common, and that is that they've got a sensor inside. The sensor consists of a lot of light-sensitive elements, and these light-sensitive elements usually are filtered out by wavelengths of light. So you have red, green, and blue. I'll get a little bit more into that just now. And then these light-sensitive elements, you can think of them as little buckets that's filling up. So as photons hit this sensor, these little buckets fill up, and then at some point you read how full are all of my buckets, and that is what generates the intensity for the different pixels of your image. There are certain other things to maybe just also realize. You get different types of sensors. You get global shutter sensors, and then rolling shutter. So what that means is global shutter means that it fills up these buckets, and then one shot it reads all of them in parallel. Rolling shutter is where you read them top to bottom. So if you've got a very fast moving thing, then there might be some, as it's reading it from the top to the bottom, the thing might have shifted. So the image that you get out at the end is maybe not actually a perfect representation of what you had. But doesn't matter in what form they come. They can be in a confocal microscope or something. It's just still a sensor. You get various types of them like CMOS and CCD and things like that, but they work on the same principle. They capture light and then give you some intensity representation matrix out at the end of the day. Now, how these images are captured specifically in microscopy is quite important. That there's this two things that's always in your mind. And the one is that there's a spatial component, spatial digitization, so that relates to the amount of pixels, basically the image size, the resolution. That's how finely you sample. And then there's also the quantization aspect, which is like, okay, well, what is the relative brightness that I can have? Now, that's where we will talk about bit depth, for example. So if you have a bit depth of 8, I'll talk more about it just now, then you only have 256 discrete intensities that you can capture. If you've got 16-bit, much more, but often if you've got like 12-bit, then it's like 4096 or something like that, 4100, I can't remember, 92 or something. But the point is that it's the quantization determines how much nuance, how much dynamic range you can actually see within your image, and that's sometimes important, especially if you want to segment things out. You need to know what is now one structure and what is another structure, and you need that intensity variation. But at the end of the day, as I was saying, you basically just get a matrix of intensities out. That's what an image is. If an image doesn't really exist, an image is just, to the computer, a matrix of values. So if it's a grayscale image, it might look something like this, where you simply have, let's say, x and y coordinates, and then they are just intensity values at each of those points. If it's a color image, so let's say RGB, then you would have a red, green, and blue component for every pixel. And then there's separate intensities for each of these three components. But basically, the main thing that you must always just have in your mind is that it's just a matrix of values. The image doesn't really exist. The image is a representation, a visualization that the computer makes to help you understand what is going on in this matrix of values. But when you do any kind of processing, all you're doing is you're manipulating mathematically these matrix of values. And I think that's important, that the image is a visualization, not the reality. Now, as a quick example here, if I just take this piece of the person's head, you might visualize it like this, but as I was saying, this doesn't really exist. All that really exists is this matrix of values that we just decide, somewhat arbitrarily, to visualize with these different intensities. I made a brief comment on bit depth, and bit depth basically represents the number of ones and zeros, number of bits that is used to represent each intensity in your image. So at each of these matrix locations, the x, y coordinates, how many ones and zeros is actually used inside the computer to represent this intensity. Now you can imagine if I've got a small number of ones and zeros, let's say for example 2, then I can only represent 4 unique values because I get 0, 0, 0, 1, 1, 0, 1, 1. There are 4 unique values I can represent. And so I have number of gray levels, if I was to represent it as gray, would simply be 4. If I had 4 bits, suddenly it's 16, 6 bits, 64, and it goes up quite rapidly. If I've got 10 bits, it's 1024, and often microscopes capture at 12 bits, so then it's 4,000, and I forgot the exact number, but it's over 4,000 unique gray level values that you have. So what happens is the true intensity coming from your sample has got some continuous distribution of intensity, but then because of the way it's captured or quantized, it basically makes it discrete, so that you've got these steps in intensity in the end. And usually this is not very desirable, because if I want to distinguish between, as I was saying, 2 different structures, we often work with such minute intensity differences that if I don't have that resolution, then I can't actually distinguish where does the one structure start and the other one stops, because it's just one intensity. Where if I've got more nuanced intensity, I can maybe detect the edge a little bit more clearly, so that I know where the one starts and the other one stops. Just to maybe visualize this as an entire image, so if you've got a 1-bit image, you basically have 2 shades, and this is what it would look like. This pattern that you're seeing here is called dithering, where you represent shades of gray actually as a representation of this ones and zeros that's just put in different densities, and then it looks like different gray levels to your eye, but obviously it's still just 2 gray levels. Suddenly with 4 shades, we get a little bit more nuance. 256 shades, it's pretty smooth. Like our visual perception is pretty smooth. But again, the image is not really there. The image is a representation of this matrix of values, and to our eye it might look smooth. To the computer it might not be sufficient. It might not be. There might not be enough intensity levels that to the computer it might look like this. It's like, oh, this is so coarse. Now I can't actually do the proper separation between what is now the leaf and what is the background, where if it had more intensity levels, it might be able to. So don't purely rely on your eyes. Remember that there is a matrix of values behind this scene. It's just another representation of the same thing of increasing gray levels. It becomes increasingly smooth, the transition from black to white. There's also maybe just an important thing to maybe mention here is that the visual perception of light intensity is not uniform. So if I was to count the number of photons, and I'm not going to overly simplify this, but if I have 1 photon coming in, and then 2, and then 4, and then if I double the number of photons, the intensity in our perception doesn't double. It's not linear. So at let me just make sure I got this right. So if you look at this, for example, this looks like a pretty uniform gradient. It looks like it's nicely, smoothly increasing from dark to light. It doesn't seem like it has a sudden jump. But if you look at the actual intensity sitting behind it, you'll see that this first jump is quite massive. And then the jumps become smaller and smaller and smaller. Even though to us this looks like a pretty big jump still, in reality the pixel intensity is jumping only with a very, very small amount. So be aware that your eyes are not giving you a true representation of what this matrix of values are actually representing behind the scenes. And just always remember that visual perception is skewed. So when we do certain changes to our images to make the visualization more appealing to the eye, it doesn't mean that the data is getting better for analysis. It just means it looks better to our eye. So that's an important distinguishing factor just to be aware of. Okay, so we've got this matrix of values. We can decide how we want to render this in whatever way we want to make it most helpful. Now, we usually use a lookup table for this. So we would say, okay, well, I've got values between 0 and 255, for example. So let's just assign a certain intensity or color or whatever to this value that I'm seeing. And then you just simply look up the value. So in the matrix, it's got a certain value. I look up what is the color that I'm supposed to render onto the screen so that it has a certain visual perception to it. And I can decide exactly what color map I want to use here. This color map or lookup table is purely chosen to help us in the visualization. It's got no benefit for the analysis at all, for the actual quantification analysis. It's only for visualization, only for representing data, communication to someone else to say, hey, you can clearly see these structures are forming here. Certain lookup tables help more with that than others. And I think there's coming an example soon to demonstrate that. Oh yeah, something else that I can also do is you can also just decide that I'm setting my lookup table to not really in reality start at, well, I invert it in a sense. I can do with this lookup table, however, you can make it that the top part is entirely white, for example. Then you saturate your image. You can make it so that it's that what is high values is shown as a dark color or inverse. So this lookup table is, you play around with this however it helps you to visualize or understand, as long as you don't end up saturate your actual pixels, as long as the intensities inside don't actually end up all clipping and saturating, it's fine as long as it helps you for the visualization. Okay, now before I just quickly go into an example, it's important to understand this idea of a histogram. So I believe you all would know what a histogram is, but it's basically counting the frequencies of some occurrence. In this case, the occurrence would be the brightness or intensity in the image. So how many pixels in my entire image has a certain brightness? You just usually plot it quite discrete for every individual brightness that you can have. You just count how many of them there are in the entire image, and you plot a certain bar with a certain height. And in this case, you can see, for example, the majority of the pixels in this image is on the darker end, but there are some in the mid-tone and brighter regions as well. And just knowing what's going on with your histogram is a better representation of what your visual perception tells you. So again, your visual perception might say, hey, I can see everything is kind of the same brightness, and then you look at it, it's like, oh, okay, no, there is actually some variation in the histogram. So the histogram is a tool that is used alongside the image. It's not necessarily in place of the image, but it's also, we will later use the histogram to do certain thresholding. So let's say I want to cut out foreground and background, then you might want to try to find where there's one peak and maybe another peak. Let's call this another peak there. And then I just say, okay, well, let's put a threshold in between that, and then everything above that point I would consider foreground. Everything below that is maybe noise or background. So the histogram is also helpful to determine where is a good place to put my threshold. And a lot of the automatic thresholding methods just basically use the histogram to then determine automatically where is the optimal place for this type of thresholding that you want to do. Okay, so I think it would be good to quickly do an example, and this is now where you come in. So every now and again I'm going to stop and then ask you to open up an image. So for now you can just open up Fiji. Yeah, so for the most part, the instructions of what I'm going to demo and what I would like you to actually do, you'll see on the screen in the slides, but there might be some things that I quickly show that's not in the slides. So yeah, you can always ask if you get stuck with something. The idea now with these types of things, it will happen a few times in this course, in this workshop, where you open things up, you click around on the same things that I'm trying to, and if you get stuck, please ask. That is the reason why you're here, is to learn, to get hands-on with it. Yeah. Okay, so you go to File, and then Open Samples, and then you find one that says .plot.
Oh, sorry.
So this is not one of the samples that I shared. This is actually one of the bought-in sample images. Okay, just tell me when everybody's there. Okay, great. You also get. Okay, so the first thing I want you to do is you've got the image here, and then you can go to Analyze, and then find histogram. You can also just press Control H on your keyboard if you've got Windows. If you've got a Mac, it might be Command H. So this is what you then get. Just maybe also making a comment, if you have lots of images open, then you first have to select the image that you want the histogram for, and then you press Control H, for example, and then it opens the histogram for that. So it's always linked to the currently selected image. That's the image on which all the processing or whatever you end up doing will be performed. So you first select the image, you just click on the window, that's how you select it, then you do whatever analysis you want to do on that. All right, so if you've got your image now, and you see the histogram, I want to demonstrate here that with your eye you can kind of see that there are low intensities. So maybe not all the way to 0, but they are fairly dark parts of the image. But if you look at the histogram, it seems like there isn't really anything going on there. And if you look at where this peak is clearly towards the brighter intensities, this high level, gray level values. And if you just think about it logically, why is that? It's because the majority of the background is of this brighter value intensity, and that's why the majority of the voxels are there. But because there are so many of them there, you kind of lose the information or the resolution in the histogram, if you want to say that, at this lower end. So to compensate for that, you click on this log button, which then basically just takes the log of your intensities. You just calculates the mathematical log of that, and then it, because log is a nonlinear function, it then for higher intensities it just kind of keeps it relatively low, and for the low intensities it boosts them. So that's what you then see here. The log is obviously not a true representation of where the intensities are or the quantity of them. The non-log version is, but this helps you to see actually, oh wait, there is actually quite a lot of intensity going on here, and you can actually now see the nuance of that intensity as well. So for many microscopy images, what you'll see is actually just the inverse. The majority of the image is black, and then you've got very small structures on this, and then if you look at the histogram, it looks like everything is on the low end, there's nothing on the higher ends. And you say, hey, I can just cut out all of these high frequencies, I can stretch my contrast and everything in my histogram because there's nothing there. And if you just click on this log button, you see, oh wait, there is actually some information there. Now, I was mentioning color maps. So the way you use a color map in Fiji is you click on this lookup table or this LUT, and then you've got a lot of options here. I'll give you my personal favorite. It's this MPL Viridis. I believe the MPL in this context stands for matplotlib, which is a Python library where these color maps or lookup tables are actually initially where they were popularized. And Viridis is a color map which is designed to be perceptually uniform. So as I was just kind of demonstrating, our eye doesn't perceive grayscale levels uniformly. We perceive them as, I did make that point, where the actual intensity is not increasing with the same intensity that we perceive as linear. Where Viridis is a color map that was designed so that as the intensities increase, your perception of how they increase still stays linear, so that you call that perceptually uniform. There were a couple of these perceptually uniform color maps. So for example, come on, where am I clicking? If you go to almost any of these, I think the plasma or magma, all of these are an attempt to be perceptually uniform, so that you can then very clearly see, oh, okay, there are some dim spots there, and with my eye I can kind of quantify the intensity of this. If I was to switch back to gray, for example, it's some of these, like that dot, for example, is basically invisible. Like I can't really see it there. Where with Viridis again, I don't know if you can see it, but I can start to make out that there is actually a dot there. So it's a very helpful color map to use. Just to maybe mention some others, often when you image with your fluorescence microscope, you would have certain expected colors. So let's say, for example, you stain with, I don't know, a GFP signal, then it's supposed to be green. It's green fluorescence protein, obviously. So then you might want to just assign a green color map to it, just to give that perception of what was actually captured by the microscope. In reality, it's not going to be as green. It's just that this is your lookup table that you put on top of the data to help you visualize and make sense of what your different stains and dyes were in your image. And you just assign a color of green so that it helps you in the analysis to just keep track of what structure is what. Maybe just another one. Maybe just to mention this part. If I have, sorry, I'm just trying to check out there's still anyone that's. Yeah, so if you click, for example, on the histogram and then you apply a lookup table, it's going to apply that to the histogram. Well, now it says it can't actually. but the point is that you have to click on the image, the thing that you want to apply any operation to. You first click on that thing and then you click on the operation. I just want to show you this. If I go to lookup table and then invert lookup table, then it inverts it. There's also a thing where I can click apply lookup table, and that's not actually going to change the inherent intensities, so you don't necessarily always want to do that. But yeah, it's good to be aware of this lookup table. I'll probably be using it quite a bit throughout here. I'm going to go into more detail about this just now. Let me just actually check when. Yeah, I can do it now. So next thing I want you to do. Yeah? Oh, sorry.
I just wanted to find out, I don't know if we maybe didn't get to it, but would you be able to select a certain portion of the image that you want to color, and then leave the rest?
Yeah, so for the lookup table, it applies it globally, but you can crop out a section. For example, I can select that, and then I can, for example, create a crop of that, and then just, I don't know, apply a lookup table, whatever to that. But if I was to select something here and I click apply lookup table, it does it on the entire image. So you can always then, after you've maybe cut it out, made some color difference, then you can merge the two. But yeah, the lookup table is a global, it's just visually, globally speaking, what are you actually doing to your image? Yeah?
So I was fighting with Fiji when you were explaining the stuff about GFP.
Okay, so what I was explaining is that when you take images on the fluorescence microscope, you get a certain intensity out depending on what your dye is. Now that could actually be some color. It's got some variation in red, green, and blue. It's not just a fixed color. But what you end up wanting to do is you want to be able to extract just a single grayscale value from your image that you captured. But now you've got different structures that you labeled with different fluorescent proteins or whatever. And you want to keep track on your mind as to what is what and how do I go about this. So then what you might want to do is you've got now your image that you got from your microscope, but it's just grayscale levels. It's just, it's not red and blue. So then you can just go to the lookup table and make, for example, if it was with GFP, you make it green. If it was with some other probe, which is supposed to be reddish, then you just make it red. And then we'll look at how we combine colors just now. But the point is that these lookup tables are helpful to keep track of what are we working with here.
So that if it was an original grayscale image and you use the lab to show green, whatever would be GFP expressed and show green.
Yeah, if you select so you would have multiple channels, for example, in your image. Some of them might be stained with something that's red, something that's stained with something that's blue, let's say the nucleus, for example. And then you just select the channel, that is the GFP one, and you say apply a green LUT to this green channel or to this channel. There's another way we will look at it when you create a composite image, then you can say this channel should be the green channel, and then it automatically makes it green.
Okay.
But yeah, it's artificial. I think that's the important point I'm trying to make here is, as you can see, I can as easily click on any color here, and I can make it that color. I didn't make it that color. Red. There you go. So it's by just putting it in a certain color doesn't mean that it is actually red or it's actually green. It's just helping you to visualize, to keep track of what's going on. And then if you can, for example, see a green channel and a red channel overlapping for colocalization, it's going to look yellowish. It's like, okay, well, the yellow is telling me this colocalization. So the colors are helpful, but they're just a visualization tool. They're not actually supposed to be used in analysis. And there are some shortcomings to that, which we'll also get into. Okay, what I was getting at is my next point. I'm just going to apply maybe Viridis. That would be a good one to work off as a base here. What I want you to do now is go to Image and then go to Adjust and then Brightness Contrast. You can also press, if you're on a Windows machine, Control-Shift-C. But just Image, Adjust, and then Brightness Contrast. Okay. Then it opens this little tiny window here. And what you should hopefully see is that this is just showing you the histogram at the top there. But what you've got the ability to do now is I can change where the minimum and maximum values are. So if I was to look here, for example, I can see that, oh, okay, the value, you can read it off here from the histogram, the value at, what's it now, around 60-ish. There's nothing really below that. There's no data there. So I can then just shift this bottom value up to about 60, and then I know I'm not really throwing away any information. There's also not really much going on here, so I might as well just saturate the image a little bit and then get rid of all of these high intensities. So that's at roughly 219. I can, again, read it off there at the bottom. I just hover over the position in the image, and then it shows me the intensity at that position. So let's just make it about 220. I can drag this down to 220, and I can see where it is at that value at the top there. And now I suddenly have a lot more visual perception dynamic range going on in my visualization. I haven't changed anything in my data. It's still the exact same data. It's just visually in my perception, I now have a much more variation between all the different structures. Yeah.
So by altering that, you want to alter the pixel intensity.
Exactly. Yeah.
So you can play around the pattern of values and still be.
Yes.
The same.
They will be exactly the same. If you click apply here, that's a dangerous button. Then it will actually change your intensities. Generally, don't click that button. There's very few cases where in an analysis it makes sense. For visualization, please change these values as much as you want. Maybe just to show you another quick trick here, I suppose, is that if I was to click on this auto button here, it then tries to automatically find the optimal values. And you can see that the values that it found automatically is 62 and 212. And if I was now to relate it to the histogram, it is 60, which I found, and then it's maybe saturating a little bit more, but it's just automatically determining where the optimal places are. I can maybe just turn this up a little bit because I feel like it saturates too much. And now you can also see that, I mentioned that little dim structure there. By having more dynamic range available, I can suddenly see that dim structure a whole lot more clearly. But again, just for visualization, this is not actually changing the values, so definitely helpful to do. I think I'm going to get more, I'm going to say more on this a little bit later, but I'm going to go back to the presentation for now. All right, so what happens when you change these values is that the histogram becomes stretched or squashed, well, depending on where you started, but it's you usually stretch the histogram so that a smaller portion of the histogram then falls the entire intensity range. So then the lookup table might use, let's say, 0 to 255, but now you take your histogram initially and then you kind of artificially stretch it out so that it falls that entire width. And as I was saying, just apply is a dangerous button if you click it. Okay, now I want to just quickly shift a little bit in the direction of talking about the optical resolution and spatial resolution. It's just important to think about this or be aware of it. So optical resolution refers to what your microscope is physically capable of capturing, the resolving power of the microscope. There's inherent limitations to it. It depends on the modality, it depends on the lens, it depends on just the wavelength of light. You can't, well, there are tricks that people do with Escher-Sim and stuff, but there are, generally speaking, no way that you can capture something smaller than the wavelength of light. So there is inherent limitations to the optical resolution of your microscope. And let's say the optical resolution is capable of capturing this amount of detail. Then the spatial resolution is how you then decide, I'm going to have this amount of pixels by this amount of pixels for X and Y, and possibly Z as well. And that is going to determine what you end up having available to you for analysis. So the ideal would be to always capture at the highest spatial resolution where it makes sense. Like there's a point where there is no more optical resolution, so increasing the spatial resolution is not going to make any difference. You're just going to have larger and larger images, wasting space on your computer. And these images have a tendency to get very large. The people that we went to Genelia last year, it ended up a week-long image run produced like 120 terabytes of data, which was a little bit of a pain to get into South Africa. But yeah, so that's the idea here of the spatial resolution is actually the amount of pixels, the amount of subdivision blocks that you end up having. And you can see that if I have small number of blocks, I'm making a lot of averaging of my intensity here, where if I have larger, or let's say if the blocks become smaller and smaller, I end up having more nuanced intensities that tell me something more nuanced about actually what's going on in the sample. I think there was a link there. Yeah, I want to show you this quickly. I'm just going to take this one. I think this is a good example. So the actual capabilities of the microscope might be like that, but then if you choose a resolution too low, you can see you're actually losing a lot of information. You're throwing away data. Ultimately, if I want to count how many of these pieces are going into the structure, when you have too low spatial resolution, and then you can't, there's no way to actually count that anymore. So just be aware of this trade-off between spatial resolution and then obviously the storage space on the computer. Generally, higher is better, but if you don't need that high resolution for various reasons, such as if you capture more resolution on confocal, you're burning your sample. You're exposing it to the heat of the sun every moment that you capture it, and it's just going to die at some point. So you don't want to actually capture necessarily at the highest resolution because you want to capture along lifestyle time-lapse sequence, and you need to then do it much quicker. So you reduce your spatial resolution, but you know that there is a little bit of a price that you pay. So just all of these trade-offs, always good to consider. Yeah, and just again, if you look at this picture that we had here, the individual pixel, let's say that pixel value there, has got no meaning really by itself. I can't really make any conclusions from a single pixel value, but I can look at a neighborhood and I can say, oh, this is kind of an edge pixel, so this is contributing to the edge. That's more showing like a transition from the inside to the outside of my structure. So in the neighborhood of pixels, you have interpretation. In individual pixels, you rarely have any meaningful interpretation. So the caution is don't over-interpret your pixel values. Just a quick comment on sampling. This might be slightly technical, but it's hopefully also a good thing to be aware of. If you've got certain variation in your signal, so this is now a time-based signal, but you can now imagine if I was to take an image, I just cut a line along it, it might be that I have high and low regions of intensity. If I want to be able to capture all of these dips, as I was saying for the previous picture, all of these pieces that go into my structure, if I want to capture all of that detail, I need to at least sample more than the height of or the frequency of my dips, because if I capture my frequency, my sampling, that's the word I'm looking for. My sampling is too low, then you see that I'll capture something that looks like this, and now it seems like there's my peak and there's my dip, and this is actually how it's going to be rendered. It's going to show a dip there when there is in reality not one big peak and one big dip, but it's actually a lot of peaks and a lot of dips. So just undersampling or making the spatial resolution too low can lead to incorrect interpretations if you're not careful. Yeah, so then the Nyquist theorem or Nyquist sampling frequency requirement is that the size of the pixel should be less than or equal to half of the lateral optical resolution. So it's directly related to what the microscope can actually capture. Otherwise, you can initially see that I've got some separate structure things going on there, and then at this resolution, I can no longer see them. So I've made my point, I think. In Z, this also applies. You also want to make sure that if you capture now with the confocal, for example, in the Z dimension, just that you set your Z step size to a value that is sensible, that you actually do capture the nuance there, because, for example, if you set it too high, you might capture nice and quickly, or you end up having something which is basically useless in Z. And here this is also quite useless. This is starting to be useful. When we want to do something called deconvolution, which we might get to today, otherwise tomorrow morning early, then you need to have a certain amount of Z resolution to do the deconvolution properly, because with deconvolution there's this understanding that I've got certain intensities in one layer that's jumping to another layer. But if I don't have enough sampling in my Z dimension, I don't know where it's coming from, where it's landed in. So I need to have enough Z resolution to ultimately get the nuance I need. And there's a nice website where you can calculate for your microscope, for all of the acquisition parameters, your XY resolution. You can calculate what must my Z step be to actually get proper sampling at the end of the day. Okay, now I already kind of mentioned this, I just want to underline these points, that if you undersample, that leads obviously to poor image quality. There are artifacts that are introduced known as aliasing. Aliasing is where you usually on the edge see these dim intensity regions, but it's not that the intensity is necessarily dim there, it's trying to compensate for the fact that I'm going through some boundary, and it's trying to make it smooth. Then you have these aliasing artifacts. Oversampling leads to larger files. Great, you've got all of your data, but you might have destroyed your sample in the process, so you don't actually gain the information that you need. And it also decreases the signal-to-noise ratio. So in other words, you typically introduce more noise. If you oversample, every pixel then has the higher tendency to be noise because it's not as smoothed out, it's not as averaged out. So there are some downsides to oversampling as well. It's not just, let's just capture at the highest resolution possible. There is a trade-off here. So then in summary, and I think this might be a good place to just have a quick break, there are a lot of things that we're trying to balance when we acquire samples, which will ultimately lead into the analysis. And I haven't said this explicitly before, but it's very important that you set up your acquisition parameters with the intent that I'm going to analyze these samples. I think it's a lot better now, Lisa. But initially, when I worked a little bit with the lab, there were certain situations where it's just like, hey, cool, generate all of these amazing data, and then it's like, but you set your gain so low that I've got basically 3 intensity levels to work with. There's no data there to analyze. So just realizing what is the thing that you're trying to do with the data at the end will determine how you end up acquiring them. So if you decrease the light intensity, then usually there's a bit more noise. If you increase the optical resolution, you get more clarity. And this noise that happens here is called shot noise. I haven't actually discussed this properly, but it's also called Poisson noise, where photons don't arrive at the sensor at a nice uniform interval. It kind of arrives just randomly. And if you don't capture enough light, then there might not have been enough photons that arrived for that area that you're trying to image. So you need to set the brightness of your laser or whatever high enough so that there are enough photons that end up reaching your sensor, so that as an averaging out of the effect of what's going on here, with enough light, you end up actually seeing the structure properly. So that's a little bit of a nuance here. So these two things increase the actual visible resolution of your image. And then the other two things which changes the amount of information in the image is the amount of gray levels that you choose, the bit depth. If you choose a too low bit depth, you can no longer distinguish between structures. A higher bit depth allows you to get more nuance of what's going on here. And then increasing the spatial resolution, you obviously get more detail. You can see where all of these bubbles are, where with the lower spatial resolution, you kind of lose all of that detail. Yes?
It's biological communication sometimes, but you can't increase the intensity. So what you mentioned is like you have 3 or 4 intensity levels of 10. If we struggle with low intensity samples, would it be better than to go for a higher bit depth, so that even though we're working in that low range, we at least have 100 or 200?
Yes, definitely. Yeah, so if you can capture at 16-bit instead of 12-bit or 8-bit, yeah, it will definitely solve that problem. But it's only necessary if it is really low intensities, because the more bit depth, the larger the image is, and then suddenly, where it took 5 gigabytes for one image, now it's, I don't know, 15 gigabytes for one image. So it's always that trade-off.
Yeah, no, we as a standard we keep to 8-bit in control, but I didn't realize the end result.
Yeah. So maybe just a general suggestion, 12-bit, it's not that much larger, but it makes a massive difference. So from 8-bit to 12-bit, you go from about 250 levels to over 4,000 levels of intensity variation. So that's a very big difference for a low expense in size.